is Dr. Ashwini Kumar Anand Raut. Dr. Raut is, as you can see in his slide, title slide, he is the Director of Clinical Research and Integrative Medicine at Kasturba Health Society's Medical Research Center, Mumbai. Please remember that this is the only center in the world dedicated to research and education in reverse pharmacology. We will hear from Dr. Raut about the concept of reverse pharmacology. Dr. Raut completed integrated postgraduate education in Ayurveda and Medicine from Benares Hindu University in 1987. And since then, he is very actively pursuing research in Ayurveda. And that research he does is in the spirit of modern scientific inquiry. As a testament to Dr. Raut's commitment to research, he has a long list of projects which he successfully coordinated. To mention a few, he has conducted a DBT-sponsored project titled Preclinical Development of Herbal Anti-Arthritic Drug. Another project funded by CSIR Nimitli is on degenerative joint disease. And currently, he is among the investigators of ICMR-sponsored Product Development Center for Phytopharmaceuticals, which is quite dear to our heart, this concept of phytopharmaceuticals. Recently, he was also involved in the Ayush CSIR COVID-19 project in a capacity of joint clinical coordinator and a consultant to Ayush CCRAS Center in Mumbai. Dr. Raut has served as an expert member of various government bodies, invited faculty to several renowned medical colleges, as consultant on clinical projects, and also served as an advisor to Council of Ayurveda Credentials and Council for Ayurveda Research USA. He is also associated with several biomedical and Ayurvedic journals in the capacity of guest editor, reviewer, and member of the editorial board. Dr. Raut is a highly productive researcher, as evidenced from his more than 75 research papers in peer-reviewed journals. Dr. Raut is also very well known to CDRI because he received CDRI Award 2007 for excellence in research for his work in Ayurveda and rheumatology. And also he has received award in 2010 from Association of Ayurveda Professionals of North America International Dhanvantari Award. So with this brief introduction, let me again invite Dr. Raut, and the title of his talk is Reverse Pharmacology, Exemplified with a Focus on Arthritis and Osteoporosis. Dr. Raut, the floor is yours.
Thank you, Dr. Naivedya, for that detailed introduction. First of all, good afternoon and namaste to everyone. My slides are visible, Dr. Naivedya?
Yes, visible. You can go to the projection mode.
Yeah, I will go to the projection mode.
Yeah.
So today I am assigned to speak on reverse pharmacology exemplified with a focus on arthritis and osteoporosis. Thank you, Dr. Naivedya, for inviting me on behalf of my institute, Kasturba Health Society Medical Research Center. Now, Kasturba Health Society, as many of you might be knowing, was started none other than Mahatma Gandhi long back in 1944. Now, these two pictures which I have shown here on left-hand side, Mahatma Ji watching it through the microscope, which indicates Satyanveshana. That means finding the truth, finding the fact. That's what we all researchers are trying to do, discovering a new knowledge. So that is the vision of this institute. And here on right-hand side, if you see Rugna Seva. Rugna Seva is translating whatever discoveries you made to the community that translational research and translational medicine is the ultimate outcome of whatever research we do. So with these words of vision which our institute is pursuing, let us go ahead and see what I can cover in this today's presentation. In next about 45 to 50 minutes, I will talk about reverse pharmacology, the approach, the path, and the transdiscipline. Then I will illustrate this reverse pharmacology path with Arogya Vardhini, which went to the Kutuki Picorrhiza Kuroa, then to Picorroside 2, and now targeted indication of NAFLD, non-alcoholic fatty liver disease. Then I will illustrate arthritis, particularly osteoarthritis, with Amrut Bhallatak, treated with Amrut Bhallatak. So it is always a challenge to study Vishadravya. Bhallatak is considered as a toxic plant in Ayurveda, and it will be interesting, I thought, to share this experience with you all on this platform. Osteoporosis, again, we had a very productive clinical study on Dalbergia Sisu, which had a preclinical work done at CDRI Lucknow with the team of Asthi, and our experience of these long-term clinical studies in postmenopausal osteoporosis I will be sharing, and lastly we will summarize and conclude with the key messages. Slides are not moving. Yeah. Now coming to the reverse pharmacology and team at Medical Research Center of Kasturba Health Society. ICMR Advanced Center of Reverse Pharmacology in Traditional Medicine was awarded to KHS-MRC in 2007. And right at the inception, we had this ICMR symposium on reverse pharmacology. This was a 2-day symposium. And then subsequently, in 2011, we had a training program of 2 weeks where the junior scientists from different disciplines, from pharmacology, pharmacy, and Ayurvedic institutions, these almost 25 participants for 2 weeks participated in this ICMR workshop for training in reverse pharmacology. And at the end, since any project has to end somewhere, in 2012, we had this first symposium, Translational Research and Reverse Pharmacology. And now we have the ICMR PDC Phytopharmaceutical Center. Dr. Naibadya just talked about that. But before that, we also had CSIR Nimitli project, where reverse pharmacology path was formally accepted or followed for all the 3 projects. One was arthritis, another was diabetes mellitus, and third was hepatitis. In between, when our center was inaugurated, MUHS, that is Maharashtra University of Health Sciences, had invited us to submit a fellowship program, a 1-year fellowship program in reverse pharmacology and drug discovery has already been submitted to Maharashtra University of Health Sciences. But when this reverse pharmacology got crystallized, it was in 1997 when in Mumbai there was a symposium on malaria. And at that time, Dr. Ashok Vaidya has crystallized the definition and scope of reverse pharmacology, and I will be sharing with you about that definition and genesis of reverse pharmacology in subsequent slides. But I want to mention here that if you see on the left-hand top, this is the compilation which is commemorated to late Vaidyantar Karji. But this contains 25 selected research publications which were jointly worked on by Dr. Ashok Vaidya and Dr. Late Vaidya Antarkarji. And on left hand side at the bottom, what you see is the translation of the Dravya Guna Vijnan, which is the kind of a pharmacopoeia, Ayurvedic pharmacopoeia, and which has been translated from Marathi language to English. On the right hand upper hand, you see this is the major project taken up by Ayush, and this is the monograph primarily developed on Panchaval color for leukorrhea and other disorders, gynecological disorders. And on the right hand side, if you see Integrative Ayurveda, again, this was in 2017, we had a major almost 2 weeks program where Integrative Ayurveda symposia at different institutions were conducted, and that compilation is there in this kind of a publication. So coming to reverse pharmacology approach. Now, in conventional new chemical entity path, we all know that we start from the molecule, then we go to the mice, and then we go to the man, preclinical, and then we go to the clinical phases. But in reverse pharmacology, since we address the traditional medicine maximum, we start with the man. That means the products are already there being used in the community, but we need to understand its safety profile. That's why we go to the animal, and then we also want to understand its mechanisms of action, and that's why we go to the molecule. So it is the bedside to bench approach instead of bench to bedside approach, and that is the simple reason it is called as reverse pharmacology approach. Now coming to the reverse pharmacology genesis, its definition and scope. As I said, it was crystallized by Dr. Ashok Vaidya, who is known, in fact, or called as the father of reverse pharmacology. But what is important, how this crystallization of definition has occurred with the continuous engagement. When he was at the helm of affairs of Sibagaigi Research Center, simultaneously he was also involved in GS Medical College, KM Hospital in Mumbai, which is one of the very prominent institute of modern medicine. And at the same time, simultaneously, he was also working with M.A. Podar Hospital and R.A. Podar Medical College, Ayurvedic Medical College, where also he was involved. So this constant engagement with the modern biomedical college and the traditional Ayurvedic college, this definition got crystallized. This was not in a 1 or 2 year. This was almost after 2.5 decades. This definition is crystallized, and I want to drive your attention very carefully. And I read this definition: reverse pharmacology is the science of integrating documented clinical or experiential hits into leads by transdisciplinary exploratory studies and further developing these into drug candidates by experimental and clinical research. Now, there is a scope of reverse pharmacology is to understand the mechanism of action at multiple levels of biological organization and to optimize safety, efficacy, and acceptability of the leads in natural products based on relevant science. So there are, in nutshell, diagrammatically, if you see, there are 3 domains: experiential domain, from where you get the hits; exploratory domains, where you optimize the leads; and then with the diverse experiment, clinical and paraclinical, you develop a drug candidate. We call it reverse pharmacology. As I said, we have to start from the clinics. Then with reductionist approach, we go at different levels of biological organizations. We study them even at molecular level in laboratories. And once we identify the targets, we try to understand also the mechanisms of action. We ensure the safety. And once that is established, then we back integrate with the holistic approach and apply it, translate it to the reapplication in clinics. So in brief, if I want to illustrate you the reverse pharmacology path, it starts with astute clinical observations. That is the foundation. From there you get the hits, but those hits have to be robustly documented. Once it is documented properly and you choose the formulation, that has to be properly standardized. This standardized formulation then can either go directly to the exploratory open phase trial, or if needed, you can also go to limited phase 1 trial. As we had gone with the Yogaraj Gugulu, we initially studied the tolerability of Yogaraj Gugulu in a higher dose range, and then the exploratory study of phase 2 can be taken, like in Arogyavardhini. Directly exploratory open phase 2 study was initiated, Arogyavardhini and Punarnavadika, and we will come to that later. But from that, you get the clinical leads. With these clinical leads and having understood the active dose range, then you go to the controlled clinical trial. But simultaneously, here you also do the experimental and cellular biochemical pharmacology. So these two, at this level, you have identified either the clinical targets or biochemical targets. And once you have identified that and you have got some insights in understanding the mechanisms and the safety profile in different animals are ensured, then large-scale clinical trials data which coming out of that, this mechanism of action, essential toxicity studies will give the drug dossier. And once that is ready, the drug for ethical promotion is allowed. That goes to the market, and again, this marketed drug generates the experiential data, and the cycle continues. So to explain it in a more robust way, I try to explain this diagrammatically. Reverse pharmacology, a transdiscipline. Now, why do we call it as a transdiscipline? Because you see, it is the experience which leads to experiments, and then we create evidence. So we get experiential hits either from traditional knowledge system or from observational therapeutics, or it may happen serendipitously. But to get that serendipitous observations, your mind must be prepared for that. So from this, we get a hit. Then we try to develop initial exploratory study, simultaneously explanatory study, and then large-scale clinical trials. Once that is done, then the evidence is generated at different levels of biological organizations. But what is important is translating this research to the community. That translated medicine for community and clinical practice is important. So this translational research is equally important. And once we develop that, again, the Ayurvedic pharmacology epidemiology approach, we can again generate a database of experiences, and then this circle continues. So why it is transdiscipline? Because it addresses at level of traditional systems of medicine and observational therapies on one hand, then it also addresses at the level of experimental biomedicine and drug discovery processes. And once it goes to the community application or in the community or clinical practice, then again, the aspects of therapeutics which community is following, may it be a dietetics or nutritional aspects or the lifestyle aspects which community is following, and it differs from region to region. So all that three domains, it encourages the interface, and that's why it is a transdiscipline. But I like to call it a dynamic self-perpetuating transdiscipline because this continuously would grow. One thing you have to remember here that many a times we go with the technology driven, and sometimes if you see in traditional medicines, we are too much personalized protocols. Like we have Prakriti protocol, then Dhatu Sartha, then Agni Pariksha, then Koshta Pariksha, and so on and so on. I remember Prakriti Pariksha, there was one protocol of more than 230 points. How it will be practically relevant? So we need to strike this balance of technology as well as personalized medical approach and personalized medicine protocols. Similarly, there need not be a drug-centric approach. We need to understand when the medicine is given or prescribed to an individual, they are having their own lifestyles. They are having the environmental impacts. They are also having their own life, their diet culture. So all those factors, when we consider, we have to strike the balance of pharmacology and therapeutics. So that is how we like to call it as a transdiscipline. Now coming to the Arogyavardhini, to Kutki, to Picooside, to targeted indication now of non-alcoholic fatty liver disease. Now Arogyavardhini to Kutki to Picooside 2. Now Arogyavardhini, it is a herbo-mineral, metallo-mineral formulation, which has got 13 ingredients, which is preferred for jaundice and which is often used in combination with Punarnavadi Kwat. Now Punarnavadi Kwat again has got 8 ingredients. So this is the intricacy and complexity of using or studying the Ayurvedic formulations. Now in long back in 1976, there was an epidemic like we have pandemic now. There was a big epidemic in at least in the state of Maharashtra and Konkan. And during that time, this combination of Arogyavardhini and Punarnavashta Kwat was extensively used clinically and experientially documented. So with this clinical use and experiential documentation, the first exploratory clinical study of Arogyavardhini and Punarnavadi Kwat was planned in viral hepatitis. Now here you can see on this slide how there is early drop of transaminase levels. Okay, within a week in these subjects, this SGOT and SGPT drop, and then it continued to drop. So the total duration, usually it is 3 to 4 weeks, but it reduced to even lesser than that. But with this kind of a lead through this exploratory clinical study, double-blind randomized placebo-controlled clinical study was planned. And as it is said that it was probably the first of its kind, the double-blind placebo-controlled study in the history of Ayurvedic drug research. This Arogyavardhini in a dose of 750 milligram 3 times a day was given in acute viral hepatitis. And that also showed a very good response, in similar kind of response in liver enzymes and reducing the total duration of disease, and also clinical features were improved much better. You can see this, the comparative study, which is demonstrated graph here. Within 7 days, as it was in earlier study, the drop was much better in Arogyavardhini group than the placebo group. Now, Arogyavardhini 50% is Picrorhiza Kurova. And on that logic, the next double-blind placebo-controlled study was planned on Picrorhiza Kurova, 375 milligram 3 times a day in again very defined acute viral hepatitis condition. Now, what was most important during this study was product standardized for Picooside 1 for its content and Picooside 2 for this much of amount. And that has been well demonstrated with phytochemical analysis. Subsequently, Experimental animal study was also undertaken with galactosamine-induced rat model, which showed reduction in HDPT, HGOT, and most importantly, also liver lipid content. So although this was reported in 1996, these two studies were conducted much earlier. We are fully aware that Kutki, way back in 1996, our late Dr. V.N. Pandey, we lost him last year only, in his Doctors of Ayurvedic Medicine. That time the degree was DAYM. His thesis, he had extensively taken the diverse experimental studies as well as clinical studies on Kutki. But later on, he focused on wider range of plant-based studies in liver disorders, and that was his PhD thesis, which was submitted in 1979. What I want to emphasize here and draw your attention to that these studies has ensured the clinical safety, efficacy of Picroruza curua, which was standardized for those actives in acute viral hepatitis, and the hint for steatohepatitis was drawn. Subsequently, when we moved to MRC KHS, a student was assigned this study in vivo model. An experimental model was developed of non-alcoholic fatty liver disease. And we all know the problem of non-alcoholic fatty liver disease, how it is emerging as a major epidemic problem, not only in our country, but entire world. So this model was very right to be studied at that time, but to develop that model was also not easy. A rat model of NFLD, which was given high-fat diet, almost 30%, was given 2 weeks of NFLD initially, and then further 4 weeks with treatment it was given. So totally it was a six weeks duration. First 2 weeks, all the 6 groups received the diet, high-fat diet, and then 2 groups received 200 milligram and 400 milligram respectively of hydroalcoholic extract of Picroruza curua, which was again standardized for picorucide 1 content, and also there was a positive control of silymarin. So what were the results? Picroruza curua regressed several features of NAFLD. Importantly, lipid content of the liver tissue, and you can see from this graph the vehicle, high-fat diet, silymarin, Picroruza curua, at 200 milligram dose and 400 milligram dose Picroruza curua. So how much it was much better than silymarin, and obviously highly statistically significant as compared to high-fat diet, or even to say the control. There was morphological regression of fatty infiltration. Now this picture is very much evident. These are the normal tissues. This is the NFLD fed rats section. This is the silymarin, and this is the Picroruza curua. Besides this, they also demonstrated hypolipidemic activity of Picroruza curua and reduction of cholestasis, which was demonstrated by reduction in alkaline phosphatase. So all these features encourage the group to go further and plan an in vitro study of model of NFLD. And this study was done by our PhD and postdoctoral scholar, Dr. Hiteshi Dhami, under the guidance of Rama Vaidya and Ashok Vaidya, sir, and also in collaboration with NDRI Hyderabad Diabetes Center, and also TIFR at Mumbai. So hepatic steatosis was stimulated in HPG2 cells using oleic acid and palmitic acid. Now this study was done in kind of 2 phases. The initial phase, as we all know, NFLD increases the hepatic free fatty acid uptake, lipid synthesis, there is an impairment of beta oxidation and then a decrease in lipid export. And on another side, oxidative stress is induced and mitochondrial dysfunction plays a significant role for the progression of non-alcoholic fatty liver state to non-alcoholic steatohepatitis phase. So NFLD is a wide spectrum, and in some subjects, the spectrum from traveling from the NFL to NASH to the cirrhosis may take a longer duration, and in some individuals, there can be a very fast progress. So that these two kinds of phases of study, in early phase study, what was demonstrated when the cells were pre-treated with picorazide 2, it attenuates uptake of hepatic free fatty acid by decreasing the expression of FATP5 genes. It also showed that it reduces the expression of this transcriptional protein and SCD, which highlights its anti-lipogenic activity. It has also shown that it reduces the expression of gluconeogenic genes, FOXO1 and PepsiK significantly. Now this is very important because we all know that NFLD is primarily seen concomitantly with metabolic syndrome, obesity in particular, and insulin resistance type 2 diabetes, hyperinsulinemia at the first stage. But with this activity demonstrated, it has a tremendous scope of mitigating this obesity and hyperinsulinemia and type 2 diabetes. In this second phase, Dr. Hiteshi has demonstrated that there was decrease in reactive oxygen species formation with increase in expression on manganese SOD and catalates. It also showed that increases the level of total glutathione and also the ratio of reduced and oxidized glutathione. This again indicates that it has got a strong antioxidant properties. Increase in expression of cytochrome C, metabolic mitochondrial membrane potential, and ATP generation again indicates that it tries to regularize the dysfunction which is known to be occurring in this model of NFLD. So this was really eye-opener that single active derived from Picorrhiza courroa like picoroside has got this multiple level action attributing to its tremendous potential to play a role in NFLD as a spectrum. Now, this is an excellent graphical representation Hiteshi has provided. Here, this is the NFLD, where these are the different biomolecules and how in this mitochondrial cytosol they are impaired. And when it is given or pre-treated with picorazide 2 in this in vitro model, how all these molecular level biomolecules get corrected. So it was a very nice understanding the disease activity at molecular level. So coming to the Arogyavardhini to Kutki to picorazide 2. Now this was the side we had seen how the team at MRC KHS has worked, but we are fully aware that other places also Arogyavardhini Kutki has been worked out. There has been diverse experimental studies on Picorrhiza courroa extracts, and its fractions have been reported not only for hepatoprotective activity, but for various other biological activities such as anti-inflammatory activity, anti-arthritic activity, anti-diabetic activity, anti-asthmatic activity, collagen synthesis promoting, and collagenase inhibitory activity, hyaluronidase inhibition. Now, all these activity are indicative that its role is tremendous in musculoskeletal disorders, particularly inflammatory arthropathies. Now, even without knowing these insights, I have been using Arogya-Vardhini in rheumatic disorders with very dependable responses. Besides this, immunomodulatory activity, anti-carcinogenic activity has been demonstrated, antimicrobial and anti-leishmanial activity has also been demonstrated. There are few clinical studies with Arogya-Vardhini alone or with Picorrhiza courroa alone or Picorrhiza courroa putki in combination with other medicinal plants in river-related and other disorders has been investigated with various outcomes, variable outcomes. We know iridoid glycosides like cutkin, cutkosides, picrosides, cucurbitacin, triterpenes, and simple phenols are such as apocynin are most explored phytoactives of Picorrhiza courroa for their biological activity and potential clinical applicability. But there are we may not be aware that 185 phytoactives have been identified so far from this single plant. And we are all aware that CDRI since 1990s has undertaken extensive study on Picorrhiza courroa for hepatoprotective activity, and the standardized product Picorrolive is developed. This Picorrolive is now been formally accepted and approved by DCGI CDSCO as a phytopharmaceutical. To my knowledge, probably this is the first of the phytopharmaceutical approved formally by DCGI and CDSCO. So Picorrolive, a standardized fraction, is now ready for the phase 3 multicentric randomized double-blind placebo-controlled clinical study. Now these multi-centers are from All India Institute of Medical Sciences, PGI Chandigarh, Hyderabad Nizam Institute, from Lucknow, the clinical center that the known prominent medical college, then from KEM Hospital, Mumbai, and also Dr. Sarin's Institute, that Institute of Hepatic Biliary Research. So all these 6 centers are working together, and it has been sponsored by ISMR, PDC, and KHS-MRC is an important partner. In fact, we were the proposer of this kind of Picorrolive or Picorrhiza courroa extracts to be explored in NFLD, and then we have joined hands with CDRI Lucknow and all these centers. So finally, friends, reverse pharmacology, as we know, it has a prerequisite of authentically and standardized product. It has to be formally regulated and should not be taken as a nutraceutical or a dietary supplement, and it has to be scientifically validated. But what is the scope of reverse pharmacology? I can illustrate it here. It can have either traditional formulation, traditional extract, non-traditional extract, bioactive fractions, active principles, or analogs and derivatives. Now, Arogya-Vardhini, as we know, is well standardized, available. Earlier also people were using, but now the doses and the dose range is well defined. Many traditional practitioners are using it very confidently. As I said, I am using these for rheumatic disorders. Putki extracts are now available in many herbal medicines. Most of the herbal medicine available in our country, at least, Kutki is one of the important ingredient. And now we know the phytopharmaceutical Picorrolive is already developed with specific bioactive fractions. There are almost 8 bioactive fractions with identified quantity of Picorroside 2, Picorroside 1, and many others. And then you will be surprised to know this Chinese group is now have succeeded in developing the synthetic Picorroside derivatives. They have almost identified and developed 12 such phytomolecules, I mean synthetic molecules based on this scaffold. And out of that, 6 have already shown the similar activity of hepatoprotection. So friends, when we are at the Arogya-Vardhini using clinical, there is a scope of diverse utility, as I said, and the safety is much higher. But as we go to the molecular level, we certainly compromise the safety profile. But we definitely have more precision and specificity, which we lose when we use the multi-ingredient complex formulations. Now coming to the arthritis, we have focused in osteoarthritis through the Nimitli, as I referred to, CSR Nimitli program. So in arthritis project, we had focused our work on osteoarthritis, and in osteoarthritis, we had taken, our team from KHS-MRC had taken lead in Amrut Bhallatak. And why this was given or assigned to us? Because it is always challenging to study the Vishadravya. This is considered as a Vishadravya, that means a toxic plant in the list of Ayurvedic medicine or the Ayurvedic pharmacopoeia. And that's why it was very interesting. I thought that I should be sharing our experiences with to this audience. Now, Amrut Bhallatak, preamble of the study. Now, as I said, this was CSR Nimitli sponsored program, where we had a brainstorming before selecting even the classical Ayurvedic plants. So there was brainstorming amongst the Ayurvedic experts, rheumatologists, pharmacologists, and biomedical scientists. And we shortlisted 10 plants, and finally Bhallataka and Guduchi were selected owing to its Ayurvedic plausibility and biological plausibility. Now, we know Bhallatak is a Vishadravya, it is Pitta aggravator, and also Rasayana, that is rejuvenator. Similarly, Guduchi is adaptogenic, Tridosha Samak, but it is also a Rasayana in a different perspective. So we selected a formulation having both these ingredients called Amrut Bhallatak Avleha, identified from the classical Ayurveda literature. Now this was also a quite exercise. There are 4 types of Amrut Bhallataks, and we had to choose an Amrut Bhallatak which will be suitable to develop as a product in this for the study. So it was an informed, intuitive choice. As I said, it was chosen from Bharat Nigantu Ratnakar, which had Bhallatak and Guduchi 40% each, and 25 other prescribed dravyas were there. So pharmacognostic and chromatographic patterns were confirmed, and raw material was standardized. Amrut Bhallatak electuary, which is in the classical form, was transformed into convenient dosage form of tablet. Purity and safety studies were ensured. Pharmaceutical standards were taken care of the tablet, and then acute animal toxicity study was also ensured for the safety. In vitro study was also conducted, which has demonstrated inhibition of hyaluronidase enzyme and nitric oxide release. So product standardization and preclinical safety activity was demonstrated. So this was an open label 6 weeks clinical study. 45 patients were enrolled based on the SCR criteria of osteoarthritis knee. Dose escalation every week. As I said, this was again a new kind of fixed flexible dosage regime suggested by Ashok Vaidyaji, a novel approach which was inspired from the Vardhaman dosage regime of classical Ayurveda. Special protocol and CRF was studied of Ayurvedic formulation without compromising GCP ICH guidelines. Special instructions such as Aushadhi Kala and Anupana, Aharapatya, Viharapatya, and then exclusion of exclusive Pitta dominant Pikruti were excluded. That kind of care was taken because it is known that Pitta dominant Prakruti will definitely have an adversity when it is used, Bhallatak particularly. So exploratory, this study, the design, as I said, the tablets were given in an incremental dosage fashion, 1 tablet 2 times a day for first week, with a dose escalation every week to 6 tablets 2 times in 6 weeks. These were the primary measures, pain was and WOMAC index, and also the secondary assessment measures were walking time, paracetamol consumption, patient and physician global assessment, health assessment questionnaire, and also Prakruti and Ayurvedic evaluation. These are the representative graph. You see these 4 lines, they are basically the pain was scale, where the pain at rest in both the knees and pain on activity was assessed. And this is how you see the typical relief of pain by 4 to 5 weeks. And then there is a complete relief of pain. This was a 6-week study. So after 6 weeks, the drug was stopped. And more than 75% patients had shown a dramatic response as far as the pain was concerned of osteoarthritis. And then, although the study was completed, we continued observing these patients even after completion of this study. And what we observed, that at least minimum 3 weeks, there was no recurrence of pain in all these patients. Some patients have gone almost to 3 to 6 months. There was complete relief. And what we found, that these are the representative graphs of 2 patients, that after 3 months, when they got again pain, this was the earlier graph. After 3 months, the patient got the pain again, and we introduced these medicines to them. And what happens, there was again similar kind of relief within 6 to 7 weeks. This is another graph. Again, after 4 to 5 weeks, the response was demonstrated. So what is the implication? It indicates, infers that Bhallatak based formulation was well accepted, formulation was well tolerated, on gradual dose escalation and on adherence to the Ayurvedic specific instructions, that is important. Asymptomatic rise in liver enzymes in 3 out of 45 patients with higher doses beyond 5 tablets twice a day was seen. Substantial relief in pain and VAS and WOMAC score was demonstrated beyond the dose of 3 tabs twice a day. Therapeutic window was identified between 12 grams Bhallatak to have an activity. So there was a narrow therapeutic index. This is a toxic plant. With the narrow therapeutic index, it was a challenging task. And then what was interesting, that persistence of effect, and then reproducibility of this response. So pain relieving activity and disease modifying potential was seen in this product. And that is how a long-term study was planned of 6 months. In this, what we have decided in this long-term study, that we had to compare it with the glucosamine sulfate, the highest sold natural product for osteoarthritis. And then response variation in these 2 different dosage schedule of FEFN02, incremental dosage schedule, as we had done in a phase 1 kind of a clinical study, and then continuous minimum dosage schedule, as we will be giving glucosamine sulfate in a continuous dosage form, daily basis. One group was given continuous minimum dosage. So this was, as I said, glucosamine sulfate comparator randomized control study of 24 weeks in 112 patients. These were the fashion in which the study was given, as I said, continuous dosage, and then pulses, 2 cycles, drugs and no drug phases. That is 6 weeks drug, 6 weeks no drug, 6 weeks drug in an incremental way. These were again the pain VAS and WOMAC secondary efficacy measures, as I said earlier. But importantly, urinary CTX2, TNF alpha, SR1 and SR2, and MRI in selected cases were also studied. So this was a double-blind placebo-controlled, not always applicable because of this design. But then clinical evaluator was kept blind. So these are the results. You can see here the glucosamine group, Amrut Bhallatak phase, one group, and Amrut Bhallatak second group continuous dose. The number of events were equivocal, and the number of patients were also equivocal. Amrut Bhallatak in osteoarthritis, if you see, the adherence to Amrut Bhallatak was much better to even to the glucosamine sulfate. So the toxic plant-based product versus over-the-counter natural products, if you see this formulation of Amrut Bhallatak, patients had adhered to in a much higher percentage. This was the response in the total pain VAS score. You can see here although there was no much statistical difference, all the three groups responded very well. ABFN02 was slightly better than glucosamine sulfate. In literature, if you see, glucosamine sulfate usually responds to 40 to 45% of patients. But here, surprisingly, in our study, maybe because of the other precautions for Amrut Bhallatak which we had applied, like Anupan and other timings, probably that has also helped for improving the activity of glucosamine sulfate. So this is again the quantum of pain relief. If you can make out this Amrut Bhallatak groups, the quantum of more than 50% response was much better in Amrut Bhallatak group, 65.7% than glucosamine sulfate, 53.8%. This is again a WOMAC score. Again, no statistical difference, but Amrut Bhallatak group had an edge over glucosamine sulfate. So the observation secondary efficacy outcome, they were comparable in both the interventions. Clinical ADR profile were also equivocal, no serious adverse events demonstrated. Laboratory safety was ensured. There were normal limits in all the three groups. But again, if you see the rise in liver in transaminase levels in ABFN0 group, and this was especially more, 8 out of 39 in pulse dosage schedule, and most of them in second phase of pulse dosage. Although they were reversible, it again indicates the description of Bhallatak and the caution given in the classics. Use carefully for Pitta dominant person. So aggravation property of Bhallatak of Pitta aggravation prevails. To conclude, ABFN02 had shown significant efficacy in osteoarthritis knee with no significant clinical adverse events over 6 months. Adherence to treatment and quantum of response better with Amrut Bhallatak than glucosamine sulfate. Amrut Bhallatak has potential disease modifying activity. Asymptomatic rise in liver enzyme, although reversible, suggests need for medical supervision, especially in pulse dosage regime of Amrut Bhallatak. And Amrut Bhallatak demands further investigation for specific phytoconstituents of activity and toxicity. Now, this is again, subsequently, we were given a special kind of project through this Hyderabad National Institute of Nutrition, where again an animal study was conducted, where this kind of acute toxicity study and long-term toxicity study of 6 months duration was taken, was done. And surprisingly, you will see that this traditional therapeutic dose was safe, and there was a mortality reported otherwise in other groups to the extent of up to 50%. So again, the importance of Anupan gets underlined. So what are the determinants of therapeutic response? Dosage form, a compressed tablet, dosage regime, fixed flexible dosage regime, dosage schedule, giving it before certain time and after certain time, vehicle of administration as an Anupan, milk, and diet regime, behavioral regime, and of course the dose and duration. So all these determinants was important and significant in the outcome of this study. Now coming to the osteoporosis, Dalbergia Sisu, which we had jointly worked with Dr. Naibedya's Asti Institute of CDRI, and I am going to share our clinical experience of this plant. We know that Ayurvedic pharmacopoeia, this drug is, although explained, it has never been clinically practiced, neither its Ayurvedic rationale would suggest that it should be used for osteoporosis. But since we had a strong evidence through CDRI about its phytoactives and also the preclinical evidence of fracture healing as well as osteoporosis, we felt, although skeptical because of this background, we took this challenge of undertaking a clinical trial of 1 year duration. And in this 1 year open label prospective study, it was conducted in 30 subjects, and the age was between 45 to 69 years. It was given in a 300 milligram twice a day. We had ensured that their daily requirement of calcium and vitamin is there, vitamin D2 is there. They were assessed basically every 15 days in first month and then every 3 months for 1 year. DEXA scan of spine and femur was assessed. Alkaline phosphatase, TNF-alpha and HSCRP was assessed. And for safety, obviously, the organ function test and ECG was taken. But what was more important, that baseline symptoms and adverse events were assessed at every visit and recorded meticulously. And you will see the results and the outcome of this. Now, obviously, these were the inclusion and exclusion criteria. I won't spend much time on this, but we had ensured that menopause at least of 1 year duration, bone mineral density more than 2.5, but not more than, but less than 4. Vitamin D was ensured that it will be at least more than 15 nanogram per ml. Hemoglobin should not be less than 10. We had avoided the subjects who were frail. That's why the age group beyond 69 was not taken. We had also ensured that they were not taking Ayurvedic Rasayan drugs in previous 3 months, and other obviously HRT, anti-osteoporotic drug, glucocorticoid, and so on were excluded. So out of 82 subjects which were screened primarily for DEXA scan, and 32 got enrolled which were satisfying the inclusion and exclusion criteria, and 30 subjects were ultimately initiated and 25 completed. So you can imagine adherence to the almost 80% over 1 year for a silent disease. It was a phenomenal adherence to the clinical trial. Now, as I was referring to the baseline symptoms, what was most significant, these were the different baseline symptoms in that frequency they were occurring, pre-treatment, post-treatment, and these are some of those which were not reduced after treatment. But what was more important, that musculoskeletal cluster was the most prominent. And as I learned recently, Dr. Naibed recently published paper from your group that you have also explored this product of Dalbergia in osteoarthritis. And there are many positive leads you have demonstrated in this recent 2022 publication. So it will be very nice to note those things. So coming to the alkaline phosphatase, which is now formally accepted as a bone turnover marker, you can see there was a statistically significant drop from baseline to the visit 7. TNF-alpha, which is an osteoclastogenic marker, which has also shown very statistically significant drop from 12.04 to 2.35. HSCRP has also shown a good response from baseline 6.1 to 3.9. Now this is very interesting. Bone mineral content percentage. This was, we could do in 25 subjects. If you see this first row, there was no change. Now no change is a good sign, in the sense we all know, and it is very widely accepted, that aging per se in postmenopausal women without medication leads to steady bone loss of 1 to 3% per year. So there was no loss. Of course, some of the subjects have shown increase, but decrease was much less, and majority there was stability was achieved and the loss was prevented. If you see this Ayurvedic assessment, similarly of in 25 subjects, no change was observed. We had assessed this Asti Dhatu Sarata. Asti, as you know, the bone, its quality, nail, hairs and teeth. These are the Upadhatus of Asti, and systemic manifestation of Asti Dhatu Kshaya has also been described. So all these protocols were standardized at our end, and we observed them meticulously. And what we noticed, that there was no change in Asti Dhatu Sarata of almost 15 subjects. In fact, 10 subjects they had shown improvement. Nail also 16 subjects, hair quality, teeth quality, and also systemic manifestations. Almost no deterioration except one in hair. So what I want to emphasize here, Ayurveda sensitive protocols have demonstrated early signs of recovery. And if you fine tune these kind of protocols with more objective parameters, I think that can become a very great tool beyond much before we can understand, as we saw in BMC content. If you see these results, these are indicative or more sensitive protocols. We need to fine tune and bring in more objectivity to these subjective protocols. Of course, Prakriti was assessed, and where Vata Pradhan Prakriti in out of these 30 subjects, majority were Vata Pradhan, then were Pitta Pradhan, and then least were Kapha Pradhan. So the key outcome of this Delbergia Institute 1-year study in postmenopausal osteoporosis demonstrates the baseline symptoms of musculoskeletal disorders have substantial response. Alkaline phosphatase has reduced, TNF-alpha, HSCRP have significantly reduced, and bone mineral density content has also shown the improvement. So 1-year organ function safety, there was no significant adverse drug reaction. Sugar fasting, total cholesterol,
I hope you are almost towards the end, right?
Yeah, I'm almost within 3 slides. Yeah.
Right, okay.
Decreasing trend. So there was also decreasing trend in these lipid markers and improvement in Ayurvedic biomarkers. These are just to share with you the Ayurvedic determinants of therapeutic response. They may be related to the product. These are 24 determinants I have shortlisted here, but they can be categorized as the product related, patient related, disease related, and environment related. We need not have to look on all these 24 kind of determinants, but case to case we have to choose and distinguish which are countable determinants and which are confounding variables for a given product. So to summarize and conclude, the key message which comes out, reverse pharmacology, a transdiscipline, encourages interface between traditional medicine and observational therapeutics, experimental biomedicine and drug discovery processes, and field realities and healthcare practices. Astute clinical observations and documenting the distinct clinical phenomena would facilitate to catch the potential hints from the widespread subjective experiences. Traditional system sensitive protocols and application of relevant scientific methods would enhance the outcome quality of integrative research projects. Rationalization, standardization, and modernization of traditional medicine practices or the products is desirable to get patronage of global community at large. And finally, continuous communications, as we have now, and collaborations between Ayurveda or traditional medicine experts, biomedical scientists, clinical domain specialists, pharmaceutical scientists, and epidemiologists would demonstrate the promising way forward. And then I would like to thank and acknowledge, this is our group at MRC KHS, Dr. Ashok Vaidya, Rama Vaidya, Jayasri Doshi, our current research director, Dr. Shobha Udupi. This is myself, Dr. Dr. Ashok Amankar, a chemist, a very renowned chemist at our center. This is Hiteshi Dhami, who has undertaken the work of Picorosite 2. Shubhada Agashe, who had been very much instrumental in assessment of many of our Amrut Bhallatak related studies in laboratory. Dr. Nutan Nabar is currently our dean, and then other group was also very much supportive in entire this process. I must acknowledge KM group for Bhallatak study, Pune group also in this Bhallatak study, and the industries like Mayers who supplied glucosamine sulfate and Dudh Papesh for ABFN 02. Podar Hospital, of course, Vaidya Antarkarji has been our mentor for the entire study, but that time he was there at Podar, and all his team, along with these special individuals who were involved in this Arogyavardhini and Kutki double-blind clinical trial, were very important. And then CDRI, Asti, basically Naibedya, Dr. Ritu, Dr. Meeta Singh as a clinical partner. Again, her research fellow Wazahat was very sensitive to note these Ayurvedic protocol parameters, and of course the industry partner Dr. Lal from the Pharmanza. Thank you to one and all.
Thank you very much, Dr. Raut, for such a lucid presentation, and it was indeed very comprehensive with with the ideal mixture of introduction very much required for the trainees, the budding researchers, and well amalgamated with clinical protocol and clinical trial design, and finally the data which you shared with 3 different disease conditions that would be extremely useful for I'm sure to all these to this particular audience. I have one question is related to your Kutki picroriza. Now, will it be any plan to compare these response in NFLD with saroglitazar?
So you wanted to know that whether there is any comparative study of Kutki with other plants or other
No,
plants?
I'm talking about saroglitazar. You know, in 2020, December 2020, Zydus has got this DCGI approval. This is a drug they were using for type 2 but diabetes, but also for NFLD. They have got this approval. So, as a, this is basically a PPR alpha gamma kind of a agonist. As a small molecule, as a pharmaceutical compound, a pharmaceutical drug is there in the market since early 2021 for NFLD. So I've seen you have compared it with Silimarin, but is there any plan, or do you really think it is useful to do this comparison, or your extract picroriza is good enough to go ahead with the current state of trial? It can be then given to a wider population.
Naivedya, may I come in?
Yes, sir. Yes, sir.
Dr.
Yeah,
Ashok
please. Okay, please, yeah.
Dr. Ashok Vaidya.
Yeah. How are you? Please.
Primarily, it is our plant products have pleiotropic effects, and the pleiotropic effects are an advantage. And I think that CDRI discovery and our work earlier should find its fruition, whereas PPR, alpha, gamma, agonists or antagonists all have highly specific targeted approach. And I don't think that only PPR, gamma, alpha are involved in NAFLD. So NAFLD inflammation is what we are worried about. And we have earlier, Ashwin did not show that, but we have shown strong anti-inflammatory activity of also Arogyavardhini. And Ashwin uses also in his practice significantly that arthritis also. So we are targeting inflammation. We are targeting fatty acid entry. We are also improving the mitochondrial function. So these pleiotropic effects of plant molecules is a major development we need to follow.
Thank you, sir. Thank you. But Dr. Naibedya, probably you might be knowing that CDRI, the way the picrolive has been developed, and the entire dossier of picrolive, they have done it at an extensive and a different, not only animal model, but different in vitro models. This picrolive and the components of picrolive. have been well studied.
Yeah. I'm aware of it. I'm very much aware of it.
No,
Definitely.
but I would come in here, Naivedya. I think quickly you can study with Ashwini, picroside 1, 2, and one of the other molecules which is active. PPAR is agonist active. Because
Just 1 minute. Hold on, please. Please mute yourself.
please,
Please mute yourself. I don't know who. Somebody needs to be muted. Is it Shobha Udipi?
I think it's Shobha.
I think somebody can give her a ring and
Shobha, please mute yourself.
Yes,
Mute
sir.
yourself. Mute yourself, please.
Thank you. Please, Dr. Vaidya, go ahead.
So what I think your suggestion is very interesting in terms of in vitro and in vivo effect of picorized extract in relation to PPR alpha gamma agonism, because it's a very important part which Hiteshi had not covered, frankly. We have done a lot of molecular pharmacology of NFLD, but this one aspect was remaining. And I think if we show that, then it becomes, in the second study, a comparative effect. But just now, I think silimarin is also a pleiotropic molecule.
Yeah.
It has also multiple level
Yeah.
effects.
Yeah.
So we are, and it has shown it is being used also. But I agree with you that with future in mind, we must do studies on PPAR agonism in the lab.
And another thing to Dr. Raut is that just a little correction. The osteoarthritis studies with Dalvergias issue has been done by Ritu.
Ritu. Yeah.
Yeah.
I mean,
She was the real engine behind the entire development of.
Dr. Ritu, I spoke to her and then I could not remember her name. That's why I took your name as a group.
Yeah. And one more thing about this osteoarthritis, you have done a, I think 6-month duration trial on I think little. What was the number? Population number?
112.
112. Okay. So now, has that been followed up by some multicentric bigger trial?
Incidentally, it was a Nimitli program, CSR Nimitli.
Yeah.
That the program got concluded at that level. Industry was very much in favor of going further, but I don't know what has happened. And then we have moved away currently because this plant is very, as I said,
Naivedya knows about it.
The development of plant product of Vallatak is so challenging. No other industry was even ready to develop that product. You know.
More than that, Naivedya, you know very well, when the DG of ICMR changes or the DG of CSIR changes, their priorities change also. And the Nimitli program has taken a lot of beating in subsequent DGs. And I can say that because I was mainly involved in the Nimitli program. And I agree with you, we should pursue it further because the results are amazing and they are disease modifying.
Very interesting. Dr. Lal has to say something, I think. Please, Dr. Lal.
Actually, Picroza Crow has a very good activity, but unfortunately, it is very unstable. Almost what we saw is 50% reduction in picroside 1 and 2 at room temperature.
Within how many?
Within a year. Within a year, nearly 50% goes down.
Because this picrolive dossier, they say that at least up to 15 months, they had no reduction. That is what is reported and it is submitted to the DCGI.
Actually,
I'm talking on that basis.
There's a product from Cadila, which they have registered in Europe, and they are putting 85% overages in their product. So I'm aware because something like 15 years back, they had come to me to produce this product, and I give them a product to Cadila Healthcare. They said kid is decomposing like anything. And I'd asked a lot of other industries also. Even at that time, Chemloids was also producing this product. And finally, I told Cadila I cannot do it. And I tried to stabilize this because it has got a epoxide ring in that. So it's a very unstable product.
Means what you are saying is very correct in the sense while even planning this clinical trial, we want to ensure that it gets finished within 1 year because of this instability. But then now they have
No, but
demonstrated.
finally, how you are going to launch the product?
No, clinical trial होना चाहिए ना अभी तो बात
But clinical trial करने के बाद में you find that it is not unstable and only waste all that effort.
In that dossier, they have categorically said that till 15 months there was no deterioration.
Nothing at all, zero.
That is the statement.
Okay.
Okay.
This was my observation of our own.
Thank you so much. Thank you so much. That is your
our extract what we had done, and this is very recent at Gandhinagar when this program was there. Again, I met the lady who's heading Cadila Healthcare, and she said we are putting like 85% overages.
Okay. Thank you, Dr. Lal, for that input.
Okay, let us see if anybody else has any question, clarification, suggestion. Please go ahead.
I would like to be a bridge between those who want to launch and those who want to research. Because I have been at both the ends of researching and also launching a lot in Ciba-Geigy days. So what I feel is Chinese following up the analogs which Ashwin mentioned. And whether the epoxide entity is critical for the activity or not. And the analogs, what chemical analogs or derived thing are there, we should study those molecules, Dr. Lal, and we should see.
Yeah. Actually, degradation pattern we have to see.
Because what we did not have this strong restriction when we did the trial for hepatitis. Naturally, epidemic was short lasting. The trial was over quite fast. But you can see the dramatic change within 4 days in transamidases and bilirubin. And I talked about this with Dr. B.N. Dhawan when we started this work, and then CDRI took up also that. And the galactosamine activity of lipid reduction and the molecular mechanism shown by Hiteshi, which Ashwin discussed, make us feel that we must pursue it with full rigor of a team itself. And I think it can truly contribute to a world hepatoprotective in my judgment as a clinical pharmacologist.
Thank you. Dr. Lal, is it possible?
Probably we have to see the degradation pattern, and probably CDRI can do after degradation whether on cell lines it's working, like the degraded product. Probably that is having an activity.
Yeah, possible.
Yeah.
Let us see if anybody else has a question.
Ashwin was saying something.
No, I was just saying because this Chinese group, the reference I have got, where they have done the synthetic derivatives of picrosides, almost 1 dozen, and out of that 6 have shown the activity. Now, if that stability of these molecules, synthetic molecules, how is it? And whether there is any scope for us to develop this kind of things?
Might be. They might have done it because of the stability also. Who knows? Sometimes metabolites are more active than the actual molecule. So probably it's also getting metabolized, which is what is getting degraded outside, and that's having an activity.
Yeah, absolutely.
This was my observation, just as said, like because you're running such a big trial, so you should have information.
Yes, we should be cautious.
Then how you use it, yeah.
We should be cautious.
Okay, so I don't see any raised hand. So I will then again thank Dr. Raut for his time and his excellent presentation. It was indeed very much educative and instructive, and it would go a long way with our trainees. Thank you very much.
Thank you, Dr. Thank you.
Bye.
Thank you all.
Bye.
Bye.
Bye.